It's going to be Dr. Mark Lindsay. He's going to speak on cardiology, Myhre syndrome, clinical and bench research. Dr. Lindsay is a member of the pediatric cardiology and cardiology divisions at Mass General. He attended the University of Virginia School of Medicine, where he completed the medical scientist training program, obtaining MD and PhD degrees. He completed his residency and fellowship training in congenital cardiology and worked as an attending physician in the connective tissue clinic at the Johns Hopkins Hospital in Baltimore, prior to joining the faculty at Mass General. In his clinic, he performs genetic evaluations and provides ongoing care for children and adult patients with genetically triggered vascular disease and other familial aneurysm conditions. Dr. Lindsay is a member of the staff of the Cardiovascular Research Center at Mass General, investigating the genetic etiology and pathophysiology of human vascular disease, focusing on genetic and epigenetic mechanisms of aneurysms, dissections, and stenosis. In partnership with Dr. Lin, he helped establish a specialized clinic for the care of Myhre syndrome patients at Mass General. Welcome, Dr. Lindsay.
Hey everybody, hopefully you can hear me. Let me share my screen. Is that, can someone give me feedback?
It looks great.
Okay. Yeah, so hi everybody, I'm a pediatric cardiologist and researcher at Mass General. I started with the focus of this talk being on cardiovascular disease, but I think Angela's presented such a good overview, and I think our studies, both in humans with Myhre and of our mouse model, are pretty early on. And so instead, I thought I'd take the opportunity at this sort of inaugural patient care conference to present more of a philosophical approach to designing and how we think about therapeutics in rare disease. And I think this is the purpose of medical research, at least for me, is to work on therapeutics. And so I wanted to both give both scientists and our patient advocates some things to think about in terms of how to manage these things the best they can going forward. So with that said, my disclosures, as mentioned before, I'm a member of the advisory board and also of the Broad Bear Cardiovascular Initiative, which doesn't intersect with this work right now. So like I said, I'm gonna just give some things to think about and talk about, what does it really take to make a therapeutic in 2021? Especially for rare disease. Well, there's some really things along the way. We need to document the disease manifestations. We need to understand the mechanisms of the disease, and then there's the actual, development of a drug or a therapy. So several other speakers have touched on this. I thought, I'd be the only one, but that's great that everybody has talked about this. And we really need to understand the disease manifestations and how they change over time in order to figure out, if you develop a therapeutic, how it works. So we can document things like the arterial hypoplasia phenotype, the phenotype of small arteries, basically, both in the aorta and in, other arterial territories. And we're starting to collect these data, and we'll be able to, inform them in a more systematic way over time. We're also finding new, phenotypes, new changes like skin, abnormal healing or keloid formation. And then, of course, cardiovascular manifestations like that we really would like to prevent, like spontaneous pericardial effusions, because they can be dangerous. They are problematic medically because they're infrequent, and we don't really understand an inciting event, and therefore difficult to design a therapy for. So how do we, accomplish this and look and think about this? Well, I think Dr. Cormier-Dare and Dr. Lin have talked about this a lot in their talks, but what's really important is what we call a natural history study. I can't emphasize it enough, although I would say that there are different types of natural history studies and different levels of that you need for clinical development. But basically, this is a study, as others have mentioned, that follows a group of people over time and helps to understand how the medical condition or disease develops. Um, it's basically absolutely required in order to develop a therapeutic. And the important part about a natural history study goes beyond just documenting findings, but we need to understand how the disease changes over time. And that's the component that's difficult. We often have one-time interactions with patients, and we can write down what we have, but learning about them over time requires a lot of time and energy and collecting those records and comparing them. And in general, a large database is going to be more valuable than case reports or individual physician experiences. They're all important, but in the end, when we get to the point of wanting to test, a medication or a therapeutic, the more information we can have about the population as a whole, the more valuable it is. So again, just trying to emphasize the importance of that type of research. Well, what about understanding the mechanisms of disease? And I think I mentioned this yesterday, but what we want to understand is how disruption of SMAD4, this protein, first affects a cell and then an organ that we're interested in, and ultimately a person. And the good thing about SMAD4 is that we know a lot about it. It's not a new molecule to medicine. There's been a great deal of research on SMAD4, primarily from the cancer literature, but that helps us understand the cell biology of the molecule. As I showed this slide yesterday, we understand how SMAD4 interacts in a lot of different cardiovascular contexts, but also in other organ systems, and how the TGF-beta superfamily of molecules progresses and influences those manifestations, like cardiac fibrosis, which we've seen in human pathologic specimens. And we understand how SMAD4 influences that and catalyzes that. Again, we know the arterial issues in Myhre, at least the ones we presume are happening from our non-invasive observations, are also TGF-beta dependent and involve SMAD4 activity. So how do we in the laboratory sort of deconvolute this kind of thing? Well, we oftentimes look when we have a molecular disorder. Usually you start with trying to define what the molecular disturbance is. The good thing when trying to understand a condition like Myhre syndrome is we can focus on a single molecule, and that is the SMAD4 molecule. So we know it's dysfunctional, and therefore we can look at both it and how it works and at its next door neighbor molecules and see how they affect and how they interact with SMAD4 to ultimately manifest the disease. So when you're thinking about a therapeutic, well, are you going to be targeting the SMAD4 molecule directly? That's one approach, very direct approach. Another would be, do we identify a molecule, a nearby molecule that interacts with SMAD4 and perturbs its function? That's another time-tested way of getting an effect on your target that you're interested in. So that's, fortunately, we're pretty far ahead in understanding SMAD4 because of previous literature. So then, how do we go about developing a therapeutic? And I'm just going to talk through some steps and some considerations in developing a putative, therapeutic for a condition like Myhre syndrome. So one of the first things that we need to determine is, I think Dr. Cormier-Dare touched on this very nicely yesterday, but what aspect of the disorder, are we really going to treat? Are we going to target with our energy and our time? There's some obvious candidates, but Myhre syndrome is a multi-system disorder, as Angela just took you, Dr. Lynn just took you so nicely through, and has manifestation in multiple organ systems. So where should we be targeting? I think there's some obvious candidates. Cardiopulmonary disease is obviously probably the most likely cause of significant, morbidity, meaning disease and mortality. But other systems like the neural system may be more important in improving, day-to-day quality of life in individuals that may have either autism or significant behavioral challenges. Obviously, there are patients with Myhre syndrome who don't manifest this, but for those who do, this is an important part of quality of life improvement. So you have to make decisions because the therapeutics for these different organ systems may be different. Well, what about target identification? So this is the process by which we think about what molecule a drug is being developed against. This is a complicated process in either the public or private sector, but and that's what I was identifying before. Are we going to target SMAD4 itself, or other molecules or pathways that impinge on that function? And this is a process where we take multiple inputs before developing and deciding, on a target. Is that we take, information from human genetics, from the literature, from experiments we do ourselves, and then arrive at a target molecule to try to develop a drug against in the case of small molecules, or develop a therapeutic. And then, if you have a target, be it SMAD4 or some other molecule, what are you going to do? How are you going to target it? The approaches for, small molecule development are fairly straightforward and done all the time in both large and small pharma companies these days. They involve having an assay that correlates with your disease outcome, and then screening large numbers of molecules to find an inhibitor or an enhancer of that activity. But one thing I wanted to think about is, can instead we target the SMAD4 gene directly? I think we just passed about 3 weeks ago an incredibly major milestone in medicine. I hope everybody who's a doctor took a look at this paper. It just came out. But this is basically the first successful use of CRISPR-Cas in a human population by IV administration of the molecule to target a genetic disorder. And this was successful in targeting a disorder called transthyretin amyloidosis, which is a neurologic and cardiovascular disease where this toxic protein builds up in the body called amyloid. And in this case, the CRISPR-Cas deployment was actually used to destroy the gene, to deactivate it, more or less. Deactivating the mutant gene product decreased the amount of amyloid and the improve symptomatology in patients with this disorder. I think was amazingly free of side effects as well. It's really a striking use of technology. Well, what about disorders like Myhre syndrome? Well, the type of technology they use for CRISPR-Cas in that therapeutic was one type, but people are developing novel forms of CRISPR-Cas technology, one called base editing, in which editors can change a single nucleotide within a larger sequence, identify a mutation, and change basically that mutation within a cell. This has been demonstrated, both in cell culture many times, and now at least in one really nice study that came out recently for Lamb progeriopathy. So, there are base editors that can be deployed to change a gene change like the one we see in Myhre syndrome. There are huge challenges, obviously. This is, at this point, theoretical in terms of the basic challenges of how, and the most, the hardest part is, how do you deliver it to the target tissue? In the experiment or the therapeutic that I showed you in the New England Journal paper, that was actually targeting liver expression, which is going to be one of the easier tissues to access. But, in terms of Myhre syndrome, the question is, again, in order to target a certain tissue, we may need different delivery platforms. We need to think about, again, which manifestations are we going to target? Are we going to target the heart or the blood vessels or the brain? It depends on the priorities of the physicians and the patient population. And then obviously, although liver delivery seems to be safe and effective, well, it may not be for other target organs, and you have to ensure that kind of safety, and it's a challenge in small patient populations. Finally, well, what about a model? How do we test the outcome measure in a preclinical context? A lot of times in research medicine, we use mice because they're mammals and they're small, and we can manipulate their genomes. We also use large animals for safety studies. We have developed a mouse very similar to the mouse developed by Dr. Cormier-Dare, and I hope you guys watch her pre-recorded talk on that, which became available today. We're just starting to explore this mouse in our laboratory, but this is the kind of tool that you need, regardless of the therapeutic that you're trying to develop, to see if it works. And we are learning. We think the mouse does cause increased fibrosis in multiple organ systems. And hope to be sharing more data on this in the future Myhre Foundation conferences. And then finally, just want to touch on, well, how do you do a clinical trial in a small patient population? How do you test your drug or your treatment in a small human population, such as, as Dr. Lin pointed out, that may have a prevalence of 1 to 40,000 or 1 to 50,000. The classical trial design that we use in medicine, and really the gold standard, is what's called a randomized controlled trial, where you take half of patients and you give them a treatment, and the other half of patients you don't give a treatment, or you give a different treatment. And then you can compare outcomes in a very straightforward manner in this type of scenario. This is obviously the gold standard, and I would never speak against an RCT. It's not exactly feasible, though, in rare genetic disorders where the number of patients are very small. So there are other types of trial designs that have been proposed and are starting to be launched in rare disease populations. And these are things such as this, where you enroll a patient in a prospective natural history trial in a therapeutic trial, and very closely determine that individual patient's phenotype, and then you give a therapeutic like a medicine or whatever, and then study the effect right after on that phenotype. This type of trial design has been approved in some scenarios by the FDA to look at experimental medicines for very rare populations. So I want to end and wrap up on, well, what can a patient organization do to catalyze research and research investment in a disease with the goal of developing a therapeutic? I think a lot of people, both lay people and even physicians, have a view that the NIH funds this kind of thing, and they don't. The NIH and fund early preclinical work and discovery work, sort of discovering molecular mechanisms. But there's very little government funding for therapeutic development. And at least in the United States, this leads to a lot of funding by either small scale or large scale biopharma. And in order to interest those types of companies or entities in promoting clinical development, a patient organization can strategically invest its limited funds and to support really critical items. And those are things like I mentioned, the natural history data collection, and encourage research participation. The ability to study what an intervention does in a population is absolutely required for therapeutic development. We also need models. I'm happy to say, we have a model thanks to the Myhre Syndrome Foundation, and there's a model in Europe already moving along, and so that's fantastic. So we're ahead of the game there. And then the other thing is to steer priorities for therapeutics. It really matters, what patients and patient caregivers see as the priorities, and that can help, encourage other entities to get on board with a therapeutic development. So that's pretty much all I wanted to say. I hope in the years to come we can expand on all these points, but just wanted to get across these points in this first Myhre Syndrome Foundation meeting. And so therapeutic development requires planning and clear goals. That's easy to say and hard to do. But I think the unique etiology of Myhre syndrome really lends itself towards at least the possibility of more rapid clinical development. And patient groups can positively influence these events, especially through inclusivity and cooperativity. And that's really vital in small groups, in small rare disorders, I should say. So again, thanks for letting me talk today to the board of directors and patients and their families.